I would like to introduce you to Vincent Wang with his talk titled Reproducibility of Cerebellar Involvement as Quantified by Consensus Structural MRI Biomarkers in Advanced Essential Tremor. Wang is a PhD, is an assistant researcher, and he was a postdoc at the Montreal Neurological Institute, McGill University, after his PhD from Beijing University of Post and Telecommunications. His research interests include neurodata science, neuroinformatics, open and reproducibly neuroimaging. So, Vincent, the stage is yours.
Thank you for your introduction, and thank you for all the audience. So today I'm gonna share my experience using these fancy tools to make your research more reproducible, at least more interpretable. So okay, this work has been done in Montreal Neurological Institute with J.B. Pauling and Abbas Sedicott. So okay, let's move to okay. So what is ET? It's called essential tremor. Actually, essential tremor is the most prevalent movement disorder in this world with unknown pathology. So, and people are trying to use structural MRIs to find biomarkers for essential tremor to see what is really happened to the ET patient's brain. And when we're looking at the literature, we saw a lot of discrepancies, and I'm gonna show you later. And so what we do is we augment the small clinical data set, which is very common in clinical research, and then we are testing the sensitivity analysis for the imaging processing pipelines and also statistical models. And so okay, so why people are interested in the cerebellum? Because historical research shows that maybe essential tremor is related to cerebellar Purkinje cell degeneration, which is Purkinje cell is a one type of neuron which is very important and located in cerebellum. It connects the cerebellum with the cerebrum cortex. So let me show what we found in literature that is saying, okay, what happened to the cerebellum of the ET patients. So we saw a bunch of papers which have very comparatively small sample size, because when you've been working with, you have the experience working with clinical data sets, you're gonna know that it's really difficult to collect a large data set, especially for some very severe or rare disease. And also they have more patients. They have more patients than the control subjects, and the heterogeneity of the patients are pretty common. And people are using very different statistical methods and also the imaging processing tools. They are using different statistical models, and they are correcting for different combinations of the demographic factors. And the people usually control for the influence of the brain size, which is usually quantified by the estimated total intracranial volume or the brain volume. And but when people are looking at the sub-region of the cerebellar, people are also using cerebellum as cerebellar gray matter or the volume cerebellar as a confounder. And then they're gonna have very different results. It's when they put the effect size of all this research in one figure, we're gonna see that it's most of the studies are really unpowered, and it's really difficult to compare the results and to have a clear conclusions. So so how we're gonna try to solve this problem? So first of all, we're going to try to reduce the recent degree of freedom by doing pre-registration. Once we have our analysis plan, we're going to do pre-registration and submit our pre-registration to a journal, and then we are trying to use the use beads-based pipelines for the pre-processing and the processing, and we are collaborating actually using the GitHub, and we have the repo here we have all our pre-processing and processing and analysis codes in Jupyter Notebook there, and we want to try to solve the problem small sample size. The only way at that moment we could come up with is trying to borrow some of the control subjects from other open data sets like PPMI and ANI. But actually, when we're using the control subjects from these data sets, we need to be careful about the side effect. At that moment, what we do is we are trying to use a linear model to control for side effect, even though it's not perfect. Okay, but then we do a lot of method sensitivity analysis using different pipelines and different segmentation tools. Okay, so this is what we do. The pre-registration there on OSF. Everyone can go and check, and we have our Git repo. If you hit here, you can see. Let me see if I can show you how it looks like. So it's here. So this is a repo, and I'm currently showing you the power analysis we've been did before we that is used for our pre-registration. We need to know all the effect size from the literature and to define the expected effect size we would like to detect, so that we can calculate the sample size we need. So it's pretty easy to share between both your collaborators and all the other people who might be interested. So and last we use 34 essential tremor patients from the clinical data set, which is collected at the MNI, and also 177 normal controls from PPMI and ANI. The reason why we are using so many control patients from other data sets is that we would like to achieve a little bit higher power. So if we would like to achieve the power of 0.9 and alpha level 0.05 and effect size derived from the literature 0.61, we need more than 200 patients, and that's why we did that. Okay. And then we do a little bit data, a little bit magic, because we would like to make sure that all the covariants like age and sex and to be more comparable between the ET group and normal control group. And a very interesting thing is we almost discard all the control patients collected at the MNI because they're too young compared to the ET patients. So this is really and when I came back to China, I was working in a psychiatric hospital. I saw the same thing. So in hospitals, clinicians are really easy to collect lots of patients, but it's really difficult for them to include or recruit any control patients which have comparative like age and sex and all the other demographic features. So this is what we do. We are trying to see whether we can achieve the same results using most of the imaging processing pipelines and most of the statistical models. For example, we're using SPM to do the VBM, which is very common like 10 years ago, and we're using FreeSurfer to derive the cerebellar green matter and white matter, and we're using Shoot, which is a very common. It's almost the only software previously to get the subregions of the cerebellar, and we're trying to see whether we can have the same results from the literature using 3 pipelines and more than 10 statistical models. These are the details of the hypothesis and the analysis plan, and also the models we're trying to test. So this is the 10 statistical models. Apart from suit, which most people use for cerebellar segmentation, we also use the maggot brain, which is developed by Mallar's lab. It's called multiple atlas registry segmentation. So for their methods, they just grab some of the subjects from one of your from your sample, and you use that as a common template for all your subjects, and then they do a lot of registrations from the subjects to a template, and from template to a very nicely and manually segmenting cerebellar, which has very high resolution, and you can see that the maggot atlas provides better resolution for cerebellar, especially for the CSF between cerebellar subregions. This is way better than suit, but actually suit is really easy to use because it's embedded in the in SPM. People can just use your mouse to finish all your segmentation tasks. And these are the models we've been tested. And actually, what we do is we are using two families of models. One is a general linear model, which we use a lot, and also non-parametric models, permutation tests. We use different choices of intracranial volume control and yeah, that's all details about models. You can find all details in the pre-registration as well. So, a very important things we believe is the quality control. Of course, we can use the NIVU to do this, but at that moment we don't have such tools. So I write a small script to load the image with a specific tool and compare the segmentations together with another neurosurgeons, like one patient by patient, and this is the results we have. But actually, it's really nice to have all of this quality control, because without this, you're really not able to be confident about your results. So this is the results. And the red circle means we find that the ET group have a positive effect, which is significant if it's solid line. If you have dash line, it's a dash line with a green circle. It means no detection. So from the VBM we get nothing. Nothing is different. And for the free surfer, we found that ET patients have a larger left cerebellar white matter, and as for shoot, we didn't find anything. So the conclusion of this research is we're really not able to find a consistent detection of cerebellar involvement in advanced ET patients. And for our ET patients, it's all the pre-surgical ET patients, which means their the symptoms of these ET patients are pretty severe. And if we are really not able to find consensus in from the structural MRIs, we are really I'm really not confident that we a structural MRI or maybe the cerebellum is really involved in this disease. So okay, so this is the results of the statistical tests, and what we realize is non-parametric permutation tests are more sensitive to group differences, because I think permutation test is just like you're doing random permutation of your samples. Thus, it's naturally sensitive to the group difference, no matter where they come from. And if you're compare the cerebellar subregions while controlling for the cerebellar volume, it gives your it gives you more discoveries. You're gonna have more like p-values smaller than 0.505, which is actually, it's just a statistic. And when we look further into the results of the cerebellar subregion comparisons, for free surfer we only have cerebellar gray matter and white matter. We have some results, and for shoot we have all party results, which is really something weird. And for maggot, apparently maggot gives better all anatomically more plausible results. And it's giving some sort of like consistent results with free surfer, and also it's giving a lot of like non-significant results. So then we look further into the results of a shoot and maggot to see what really happened here. So this is a very typical results from shoot segmentation. It gives you the very nice coverage of cerebellar and also the cerebellar deep nucleus. For example, the dental nuclei you can see from here, which looks nice, but it really ignores all the atrophy of cerebellar. So you can see from the maggot results, it can capture some of the main like atrophy patterns of the cerebellar subregions. For example, cruise 1, cruise 2, you can see something between here. And when you're looking at the correlation of the volume of the subregion, shoot gonna give you a full red map, which is like about 0.9 correlation of all their subregions, which is non-informative at all. So this is a bit alarming because as far as I know, most people are when people are looking at the volume of the cerebellar or want to do the cerebellar segmentation, the first choice is shoot because it's a really nice tool. It's very easy to use, and it's actually has been embedded in most of the processing pipelines. For example, in free surfer, in FSL, or in AFNI, the default cerebellar like subregion atlas is a shoot atlas. It's something like that. It's something like this. So if you're doing registration on this atlas, you're gonna have the results like a very like fully connected cerebellar, and of course you're gonna have a like very high correlation between these regions, and this is not non-informative. But when you're looking at maggot results, it's apparently it's better. It's and also we know that from the quality control. So this is our results. But maggots, the only bad side of maggot is really computationally intensive. It's you need a server to run that. It's gonna eat up all your CPU resources, but it doesn't take too much memory. Another thing about shoot is that you need to manually adjust your brain image so that shoot can properly cut the cerebellar out for further segmentations. That's gonna take some time and also introduce some sort of like reasonable degree of freedom. Actually, I run like 4 or 5 rounds to make sure that all the subjects have been properly pre-processed. And when we're looking at the cerebellar cortical structural covariance, which a lot of people love to do, we saw some consistencies and we also see some inconsistencies. When you're looking at the right white matter cerebellum and the cortical volumes, I mean the correlation between them, the results are a bit different here. For the others, it's just like it's the sign is the same, but the magnitude is a little bit different. But actually, when we are trying to interpret such results, we should be careful. So for this work, I think it's actually sometimes it's common for the clinical research is usually your data set, the size of your data set is small, which means you don't have enough power to detect like small effect sizes. And also the cohort effect is complex, and linear model is far from enough. We need better models, and actually I don't know how to solve that, but anyway, this is something we need to work on in the future. And we know that for the clinical research, the clinical variables some of the clinical variables are very important. For example, the disease duration and the onset of the disease. But we don't have these variables in this research. And one my experience in other clinical data set analysis is that when you have such variables, it can give you always better interpretation of your data. When you're looking at R square of your model, when you have the disease duration and the disease severity, these variables, it usually has way higher R square. And also, when we are evaluating such pipelines, their performances are evaluated as the default settings, which means we didn't optimize the pipelines for the for our data set, which is also, I'm not sure whether we should do that, but actually this is also something that could introduce differences of our abilities in the research. So in summary, I didn't see the consensus between different methods and pipelines and statistical models that is saying cerebellar is involved in essential tremor. But actually, we see something. We see the red cerebellar green matter reduction, and also we and the left cerebellar white matter increase. And when I read another paper, which is a postmodern study, it's saying that in the very severe essential tremor patients, they're gonna see some of the swelling of the axons bundles in the cerebellar white matter, which might be related to our results. I'm not sure. Okay. And also, we need more open data sets of open clinical data sets to increase the power, or to model all this heterogeneity. And the results of the imaging research are highly dependent on the image processing and the statistical models. So, which means we really should be careful and about these details of the methods and the tools. Thus, pre-registration and method sensitivity analysis should be accompanied with all the imaging research, so to make sure that your results are really interpretable and comparable. And rigorous quality control is really important. That's going to give more confidence about your results. And also, there are some interesting experiences to share during doing this research. Actually, indeed, we are using Git and GitHub for collaborative coding and code review. I've been working with clinicians and also with another expert. You may know him. It's Nikhil. We spend a lot of time improve the coding, and we help each other with that. And he helped me with the code review, and it's important and highly recommended. And also the hackathons are very useful. You learn a lot from the hackathons, including the small lab level hackathons. When I was in JB's lab, we usually organize lab level hackathons. We just a bunch of guys. We sit together and we're discussing and coding together to solve small problems, and this is a really nice experience. And also, the open science community are really nice, and you can get help from the community very easily, and also you can contribute to the community easily. So after I left MNI, I go back to China, and I organize. I have been helping organizing hackathons in China as well. You can see the pictures here, and this guy is Lu Bin from he's inviting me for this talk, and we've been it's the students like hackathons. They tell us that they really learn a lot, and they would like to have hackathons to happen every year. So in China, we are organizing. Actually, we've organized 3 hackathons already, one for each year. And this is the longest hackathons in China as well. And also we are on the we're old members of the OFBM hackathon. This is the photo for 2023. So if you're interested, just sign up and join the hackathons. I'm sure there will be hackathons in OFBM hackathons or local hackathons next year. So just join and do whatever you like. You're gonna learn a lot. And thank you for your for the invitation from MRITogether, and this is all the collaborators for this research. And any questions?